Samiksha, we can start with AFSTI introduction till that time, and then maybe
Sure, ma'am.
yeah.
So, to all those who have joined, a very good morning and welcome to the Edu series organized by FSTI Mumbai chapter and CSMB, the Chamber for Advancement of Small and Medium Businesses. It is a pleasure of both of these organizations to host a series of lectures called the Food Techie Sherpas, where we come together to uh, discuss, to enumerate, to brainstorm on the various technological innovations that keep happening in the food tech and science field. We have with us Madam Ms. Chinmay Deolgaonkar, the president of AFSTI Mumbai chapter, to tell us more about the AFSTI Mumbai chapter organization and about what the series is about. Ma'am, over to you.
Thanks, Samiksha. For those who are new to the AFSTI, I would have generally asked like how many of you know about AFSTI, but since we are here for the talk, AFSTI is about 60 years plus association, exclusively of food scientists and technologists, but with an aim of bridging the gap between the industry, academia, regulators, food safety professionals, etc. And we do number of activities from conferences, publications, supporting on technical aspects. So one of the activity that as a part of our education, promoting the awareness and education on science and technology, Mumbai chapter initiated a series called Food Taking Sherpa, and every second and fourth Saturday, you will have 90 minutes talk from a techie Sherpa from the industry on completely unique topics which might not be in your syllabus. So, and this is exclusively designed for the students. And the student who attend six such sessions will be given a certificate from the association on attending this on completely different topic, which you can flaunt it on your in your CV. So with that background, I will hand it over. So I welcome Dr. Siddharth Manvati. And I met Dr. Siddharth about 3 years back, and we were in a protein-based smart protein seminar, and that's when there was interesting discussion on why plant proteins are better and animal proteins are not better. So eventually the discussion led into something like what is it that is appealing? Is it like when we are looking at the protein quality, the amino acid composition, and along with that, the number of spices and oil that we eat to make it palatable? So, what is really healthy part of it, right? So, that is how we got connected in the discussion, and we became friends over a period of time. May I request all to keep on uh, requesting all participants to keep on mute. Thank you. And so that's how we started knowing each other. And he and his beautiful wife, they are and talented wife, they are promoting the various smart proteins, including animal protein and plant protein across the globe. And they conduct very nice international conferences on the smart protein. And I'm very happy to welcome Dr. Siddharth, and he will tell the passionate story of how he started his lab, which is making this cultured meat. How he came from a scientist background and how he started his lab. Dr. Siddharth has a lab called Clear Meat. He has a company called Clear Meat, and the lab is in Sonepat. So over to you, Dr. Siddharth. We are all ears to hear your story on how you started with your lab and the topic which is of interest to all. Over to you, Dr. Siddharth.
Thank you, ma'am. Thank you very much, and I welcome all the audience today to this event. A very, I would say, I appreciate the effort that ma'am is taking, and you rightly mentioned we met 3 years back, and since then we have learned a lot from your side, and we have seen the industry grow both from the perspective of cultivated meat and from the perspective of how proteins are being identified and classified. So I think the interaction has been pretty interesting since the day we met. There have been numerous uh, discussions that we, especially you, me, and my team have had, and it has been progressive on those lines. And I feel the effort that is required to discuss or to identify the science and clear the miscommunication that is around lab grown meat or cultivated meat is much required before these products go out in the market for people to consume, because in the last couple of years we realized that there are a lot of miscommunications, misnomers, and misunderstandings when it comes to cultivated meat. And I thank AFTM for the opportunity, and ma'am, I'm looking forward for more such events from your side so that the audience gets the right perspective. Now, I will not waste much of the time, and I will simply jump into today's topic. I welcome all the audience, and do feel free to ask me any sort of questions so that we can get more clarity. That has been our approach since day one at Clear Meat, even, and my personal approach also to do more and more talking and make people aware of what things are and how it can be corrected and what is the purpose behind everything. Coming from a background of PhD in biotechnology and postdoc in synthetic and systems biology, our approach from day one as an individual was to basically identify or bridge the gap between laboratory science and its market application. I remember back in 2015 when I completed my PhD, we always used to talk internally. Yeah, we can do so much in lab, but we do not see that much market potential of it being utilized. And at the same time, there was a startup wave, and we realized we can do something out of it, and we can do something concrete for the society. Coming from a scientific background, again, we had an understanding of how food materials are made, what is the pros and cons in the processing industry, and how it can be impacted on a longer run when it comes to sustainability and other parameters of the globe. We realized as an individual and our team specifically that making meat in lab is something which we do on a routine basis. It's not specifically meat; it is growing cells in lab from a scientific perspective. So do feel free to ask questions which are directly or directly related to this topic so that we can create more clarity on that ground. Moving ahead, uh, coming to today's topic, what is lab grown meat or what is cultivated meat? Let me, yeah. Now, this topic has been prevailing in the global audience since last couple of no more than 10 years now. There has been a lot of discussions, both pro discussions, both negative discussions, and a lot to understand and a lot to grasp, but there is a lot of confusion at the same time. If you speak of what is lab cultured meat, ideally it is the same beef, pork, or chicken, or any sort of meat that we non-vegetarians eat. The only difference is that a normal meat that you order, chicken that you buy outside, is reared in farms, and what we make in lab is actually made, or what these are cells that are grown in lab without killing the animals. So end product is almost same. The process of making meat is different. Now, if you would ask me, the end product is same. The process is almost different. Now, what is the confusion out there? Now, if you look at the topic, people have identified lab grown meat as either they have labeled it as lab grown, or they have labeled it as cultivated, or cultured, or in silico, or artificial meat for say. But one has to realize that it is ultimately a mass of cells which is similar to the raw meat that we buy in market. So this is an understanding that we at Clear Meat have, that because it is a mass of cells, it can always be called as meat. It cannot be redefined into something new words. Yes, the ways of making meat are different. The ways of making these pool of cells or mass of cells is different. But the final end product will always remain the same, that is meat. So the labeling should be in the format of meat, and I don't know how the audience reacts, but would love to have more perspective from you guys also as we speak of. Now you would ask me the next question. See, it is you are labeling it as meat, and it is again lab grown, or what if the process are different, but it is meat at the end of the day. Why do you need these alternatives? What is the need for such alternatives, and why to create so much confusion and so much product pipelines in the process? There are a lot of wastages, there are a lot of resource utilizations when it comes to both the parts, and why there is an alternative. So before going deep dive into what is cultivated meat and how it is made, let us first understand why the need has actually arisen. Now, I would put it this way that I hope most of you guys might see an understanding though that 80% of humans prefer meat, that is raw meat, globally. And if you look at the data of India, in the last couple of years, 10 years back, the data said India was 69% non-vegetarian country. But now it is more than 75% a non-vegetarian country. And increase in population, there will be an increase in demand for these raw meat products. Now, as the demand rises, one has to realize what is the process of making meat and what problems it is creating. I will be pretty sure that most of you guys must be aware that out of the land space that is available in this earth, only 65% is available for agricultural purposes. And out of this 65%, up to 30% of this agricultural land is utilized for these factory farms or for rearing of these animals, which are ultimately killed and consumed. Now, these factory farms are responsible for 15% of the greenhouse emissions as we speak. Parallelly, out of the 70% water footprint which is consumable, out of this 70%, only 30% is being utilized by these factory farms for growing these or for culturing these animals. Now, apart from the utilization of the resources, apart from impacting the whole ecosystem, this not only results in land overgrazing and erosion, but these factory farms use a lot of antibiotics, and which we can see right now. There have been these antibiotics have now entered the whole food chain of humans and have impacted our immunity and have impacted how we react to diseases. There have been many cases where zoonotic diseases are caused through these factory farms. I would not go put the right names, but it is directly indirectly impacting not only the ecosystem but also impacting our routine for the coming generations. Looking at these massive problems and understanding the whole concept, that is the reason with the increase in population, there will be an increase in rise for these products, basically high protein, high nutritious products. When you speak of increasing in demand, we as an environment will not be able to manufacture or match that increasing demand as we speak of. And currently, I will put an example to this. I don't know if you guys have ever read or realized. We in India we normally go out to McDonald's. McDonald's saw an increase in demand for chicken and the supplies of chicken declined, and they gradually, slowly increased the price of chicken nugget or chicken burgers. But as there is a demand, we never realized it. And when I got across this data, I had a check on the price point of these chicken burgers. I realized, yes, the prices have slightly increased because the demand of chicken ingredients have declined, but the demand is increasing with every passing day. So that becomes one of the reasons, apart from sustainability, apart from how it impacts the nature, the reasons for identifying alternative proteins. As a manufacturer, alternative proteins becomes one of the key source to identify products based on the consumer demands. When you speak of alternative proteins, we have already this is a very broad spectrum that I put in place today in front of audience. One is the plant-based protein, and another is the cell-based cultivated meat. When you speak of plant-based proteins, it is identifying plants, for example, like soya, mushroom, and likewise extracting, doing plant extracts from through routine models and creating meat lookalikes. And when you see of cell-based meat, it is taking a biopsy, growing these cells, and making 100% meat out of it. But because of the nutritional value, because of the texture, taste, these cell-based meat, because they are now 100% meat, these are mass of cells, will always remain the preference of meat eaters. And that is what we have obviously learned in the last couple of years, how plant-based industry started, how plant-based industry gave out products. Even soya-based products are in India's kitchen for the last 10 years now. But still, meat eaters do have a preference towards 100% meat or raw meat. So we have to realize that with the increase in population, there will always be increase in demand for food, and that demand is also governed by the rise in meat expectations. There has been a study by various groups that proposes that by 2045, our plates or dining plates will look as if there would be 20 to 30% would be plant-based alternatives in our dish. Another 30 to 40% will be cell-based meat, and rest of it will be the traditional way how we eat our food products. So in the coming years, these alternatives will become an option for people to match their demand for meat or meat lookalikes, I would say. Moving ahead, as we speak of, you have to realize the process of making meat. Before I deep dive into, I may give you an understanding of what this industry is, why we are trying to do it, why the demand for alternatives is actually out there. But what is the science behind it? The process is very simple, and there are various technical issues associated and various impactful issues associated. But from a process perspective, I've taken chicken as an example to make it more understandable to the audience. The method here is identifying a biopsy or taking a chunk of the a piece, for example, a muscle. That is what we started off with. We took a muscle biopsy from a chicken, healthy chicken, without killing the chicken. We have to remember that. Without killing the chicken, we identified this biopsy, identified the multiple cell types a single biopsy has, identified these particular cells that we need to multiply to make meat in our case, and after identifying these cells, we now provided the right nutrition, which we call scientifically cell growth media, to these cells. Nutrition so that they can multiply. They can multiply and make mass out of it. Now, this multiplication then ultimately results in making the chicken nugget or chicken keema or minced chicken that we speak of now. If you look at the process, I presented a very simple, simplified way of looking at it. The process involves no killing of animal. In the whole process, there was no utilization of antibiotics. It was all in a controlled environment. The chances of inducing zoonotic diseases decreases, and we get a final product out of it. But there are certain key challenges into the process itself. And when you speak of these key challenges, we at ClearMeat actually identified four pillars of making cultivated meat. These four pillars become the number one pillar is the cell lines or the cell banks, the source of cells. If it is from a chicken, then it becomes a chicken cell line. If it is a chicken muscle, it becomes a chicken muscle cell line. If it is from a fish part, then it becomes a fish cell line. From a camel, it becomes a camel cell line. From a crab, it becomes a crab cell line. The second pillar becomes cell growth media, the nutrition provider, so that cells can multiply and grow and give us the right amount of mass that we require. And this cell growth media is normally obtained from fetal bovine serum. I will deep dive into it. The third pillar to this whole technology is the method. It can be either scaffold-based method using a base to so that cells can create those structures, or using bioreactors. These are round setups where there is a continuous supply of media, cell growth media, and cells so that cells can multiply, and then you can take out these cells. The fourth pillar, which is also an important pillar, is the licensing, the FMCG corridors, or how you do the sales and marketing and how you put it to people for them to try it out. When you look at all these four pillars, you have to understand the importance from both manufacturing as well as industrial setup that actually governs the cost of the final product. In this cost of the final product, the second pillar that I spoke of, that was cell growth media. If you look at cell growth media, I mentioned it is made from fetal bovine serum. This fetal bovine serum, going ahead or slightly deviating from food technology, this fetal bovine serum is actually the prime source for providing doing any cell culture based research. When I speak of any cell culture based research, it is vaccine production, it is drug discovery, it is doing cancer research, it is doing karyotyping, and likewise. The whole purpose of this fetal oil serum is to actually provide nutrition to these cells. Providing nutrition, by that we mean to providing them the right energy so that they can multiply. Now, there are various types of fetal bovine serum. As the name suggests, it is actually made by killing calves. So remember in the beginning I told that we make meat, the cell-based meat is actually obtained without killing animals. But the ingredient here, the ingredients play a very pivotal role in understanding the concept of cell cultivated meat and the purpose of identifying alternatives to meat from a sustainability perspective. This fetal bovine serum, I'm sure most of you guys might be knowing, only 3 to 4 countries make fetal bovine serum globally. India itself is an import only country to fetal bovine serum. Apart from the method, the process, this fetal bovine serum is responsible for 60% of the production cost when it comes to making any cell based meat or doing any cell based research. So, the second pillar plays a very pivotal role in making cell based meat or in doing any routine cell culture. Now, we have to realize what is the hidden problem here. Speaking of the hidden problem, if you look at fetal bovine serum, so there was as we spoke of 4 pillars, the 2 pillars that become the building blocks or the starting material for cell-based meat. One is the cell lines or the cell banks. I totally accept that. Yes, creating a cell bank, one student can sit for 1 year, 1.5 year, and create his own cell lines from any source without killing the animal. But you have to parallelly maintain the genomic stability of these cells, and that is the most tricky part. You have to identify or keep the cells intact for their whole passage life, that is multiplication rate. But when you see at cell growth media's perspective, this cell growth media is actually obtained by killing calves, as I told. Now the process is pretty gruesome. I took a very animated photograph of it, but the process is pretty gruesome because these cattle or cows are actually those which have been already reared and the milk has been churned out and they have been utilized to their maximum, and now they are left to die. And even at the time of death, the blood is extracted, the calf is killed within the womb, and the serum is extracted from that process and utilized for experiments or research purposes. As I spoke of in the beginning of why the need for alternative protein from a perspective of sustainability, yes, we do realize that we have made meat out of it, we can identify alternatives to it, but we need to understand the building blocks of this process, what impact they pose into the process of making meat, and how it actually replaces or fits in the vision of sustainability. On this line, if you look at the process of fetal bovine serum itself, as I mentioned, it is obtained by killing pregnant cow. It requires huge factory farms for rearing this cow. The process is highly non-sustainable. Again, because of its non-sustainability and the amounts of logistics involved, it is very expensive. And the procurement time, because of the process and demand supply, is very high. Today, if you order fetal bovine serum, it will take minimum 6 months for that fetal bovine serum to come to your place, and there are various ground checks on it, from quality to how it is being used, what is the perspective. And as I mentioned, India is a 100% import dependent country. In During COVID, if I'm right, $24 trillion of fetal bovine serum was imported for vaccine production or for routine R&D. I'm wrong on those lines, but for routine R&D. That gives you a perspective that how much important this fetal bovine serum is. So from a fetal bovine serum perspective, 20,000 cows are slaughtered every day. These numbers are raw numbers or recorded numbers. I don't know what happens on the unregulated formats. The rearing of these cows which are being killed to make fetal bovine serum, is responsible for 12 million metric tons of greenhouse emissions globally. And the land space, as I already mentioned, apart from utilizing the agricultural land for either poultry farming or other rearing of animals, this itself is responsible for 3 billion hectares land usage. And antibiotics is totally different. So when you speak of making meat in lab or identifying alternative proteins or identifying alternatives to meat, yes, plant based is something which is important, but because of the acceptance from a different note, when we did our roadshows to identify how, coming from a scientific background, it is pretty difficult for us to understand the consumer behavior. But we did a small survey to identify what all are the factors that are responsible for any food item, irrespective of its source, to get accepted by masses. When you speak of these items to get accepted by masses, there are 5 basic parameters which we identified. I might be wrong, and we are still learning on those lines. These 5 basic parameters included: one is taste, another is texture, next is smell, next is nutrition, and the fifth one is price. So, for an individual, these 5 parameters play a very vital role in accepting any food item. And if you see from these 5 parameters and the process of making this alternative to a meat, which is again 100% meat, that is lab grown, cultivated meat or cultured meat, It has the potential, but the method is still very unsustainable. Now, as I shared with you guys, and as I spoke of, from the methods that we discussed, what could have been, what we actually achieved with cultivated meat? That it might be your question when I'm myself from a cultivated meat industry, making you guys accept the process and making you realize the non-sustainable ways of making meat. So when you speak of the cultivated meat method technology that I discussed right now, yes, the one is you obtain lab grown meat. You save one chicken. For example, in our case, we made chicken keema out of it. You save one family of chicken, but at the same time, you are utilizing fetal bovine serum. Now, use of fetal bovine serum is resulting in killing one family. So the whole chain, which I totally agree to. There have been many people who discuss about the pros and cons of cultivated meat, and they question the cultivated meat industry itself, apart from how many numbers we target, apart from what impact we show. But they question the process, and I'm totally in line with them. I totally agree with them because you have to realize the purpose, the reason you started this whole industry, the reason this concept was put in place, and why everyone out there is working on it. So the method to achieve success from the lines of cultivated meat, which now most of these specific companies which have grown or become unicorn, is basically identifying alternatives to fetal wine serum. Now, these alternatives are actually used without, made without killing of animals. So, if you look at cultivated meat itself, there are two major pillars. One was the cell line, the cell banks, and the other was cell growth media. Yeah. So another was cell growth media. Looking at these two parameters, the alternatives to, once the market has alternatives to fetal wine serum, and this industry is using non-animal based, non-antibiotic based, non-synthetic chemical based methods, it becomes a more viable or a more sustainable value chain proposition. And that is what we at ClearMeat are trying to do. Our, if you look at alternatives to this fetal wine serum, the solution, as I discussed, using no kill animal free, cell free media, again identifying a cheap method because price was one more parameter that was responsible for acceptance apart from the taste, texture, smell. The reusability of media, the reusability of growth media, and how sustainable the method is.
Wow.
This is something where which I am personally impressed with how government of India is pushing it. We have seen, if you speak of cultivated meat from a global perspective, as on date, there are only three countries which have approved, I would not say approved, but they have given a no objection certificate to cultivated meat industry. One is Singapore, next is US, and third one is Israel. Now, from an Indian perspective, this is my personal observation, which I've realized that India is looking at this industry, the alternative protein industry, from a different lens. For countries like Singapore, US, and Israel, or for say Japan itself, or Europe itself, for them it is more of a demand supply problem. They do not have that much demand of alternatives or solutions or food materials to what the demand out there is. But for India, which we realized in our G20 summit also, the government of India's mandate from a circular methodology, how waste management is done, how traceability is done, personally feel the way India is slowly looking at it is more of a mature way of understanding the method. Because as I mentioned in few slides before, it is not the how these cells are grown or what instruments are utilized to make these cells. It is actually what ingredients are being used, how these ingredients are directly, indirectly impacting the sustainability format, the climate change format, and what is the final outcome, how there is a waste management to it, how there is a traceability to it. Now, these are certain parameters that other countries are not looking at. And I would share with the audience that Japan is actually following the format of India now. They are closely monitoring how India is discussing these, and we ourselves have received numerous interactions with the Japanese authorities where they have started discussing what are India's objectives on it. We at Clear Meat, we have represented India on global fronts discussing how India thinks about it. So I feel confident that yes, initiatives like these and what we are having today are much required, and that is something India is doing differently from others, from the technology and also from the marketing perspective, and is required to understand, better understand the alternative and better understand the reason for doing it and what are the impacts of doing it. Coming back to the presentation itself, if you look at, I've discussed a global perspective about what method are being adopted, what is the cell technology, how cells are grown, how these cells are multiplied, the importance of cell growth media, the sources of cell growth media, the importance of identifying replacements for cell growth media. And this is where ClearMeat has taken a lead in India, putting India on the upper hand when it comes to the globe. We already have our own in-house minced chicken. We have our own cell growth media, which is made without fetal bovine serum, does not use any animal killing or result in animal killing, is made from 100% plant extracts, the cell growth media part. This cell growth media is used to make our own meat in lab, and we have already made the meat, and we are now doing internal validations and going for industrial POCs on those lines, because we are able to make or use. And this is the slides that I discussed about identifying alternatives, identifying more sustainable ways of growing meat, and the understanding of the various pillars that are involved in making meat. When we applied it in routine setups and on industrial POCs, we realized yes, it is able to do an impact. As we are able to reuse our cell growth media, which is again made from no fetal bovine serum, no animal killing, we are able to bring down the price of our cultivated meat. And gradually, as the demand rises, obviously the price will be achieved in a price parity format. So, looking at the technology and the pillars and the expectation of the consumers, this is something that every company is trying to achieve, making more customer friendly products. Moving ahead, so from a clear meats perspective, we have been working pretty hard in the last couple of years. I think we are now a 4.5 year old company. But from a research perspective, we had been working on this cultivated meat back in 2017, 16, 17. Now, you have to realize that the four pillars that I spoke of, the cell lines, the cell banks, the first one, because we make our own cell lines from the resources that we take, we are able to take charge of the ingredients or input ingredients, initial input ingredients, because we make our own cell growth media, which is again without any animal killing. We know how it works, how it performs, and how it provides the right nutrition to cells. And the methods, again, these are all in house. So there is a catch to the methodology that we adopted. We use both. There's a reason I did not discuss scaffolds in bioreactors. I did not want to put in more science to today's discussion, because that discussion I would love to have separately also, because these are deep dive discussions from a technology perspective. Now, at Clear Meat, from an Indian perspective, if you look at the ingredients that are required or the methods that are adopted, and how government of India is pushing for circular setups, what we did is, because our Clear X9 or our cell growth media is made from plant extracts, the leftovers of those plant extracts are actually used as scaffolds for our hybrid meat. Now, in our case, we grow our own cell lines, we make our own cell banks. We are not dependent on anyone which is outside India or outside our parameter. We make our own cell growth media, which is again no animal killing, no antibiotics, and we use our own bioreactors, our own scaffolds. We are able to bring down the price of cultivated meat mostly by 60%. The initial understanding or of the method and the initial POC showcases that once we go for commercialization of our meat end product, the price of cultivated meat will be somewhere around 1500 INR to 2000 INR per kilogram, which fits in perfectly to the processed meat industry. Currently, if you go out and buy these chicken sausages, it comes at a price point of 1500 INR per kilogram. So, in gradually as the demand rises, as the method becomes more customer friendly, yes, the prices will go down, and we will be able to match the acceptance, consumer acceptance when it comes to, as I mentioned, taste, texture, nutrition, smell, and price. And this is where Clear Meat has been continuously working to create more awareness, more thoughts, and the whole agenda. See, in the beginning I mentioned about lab grown or cultivated meat being labeled as meat, but why the alternative? It was more from a sustainability perspective, more from a climate change perspective. But in that whole process, you have to realize what the methods that I discussed, the overview that I gave you today, how it actually impacts or makes sense on ground. When you speak of on ground impact, we did an analysis of how we are doing it and how our methods have actually impacted it. Because the technology that I shared does not use any antibiotics, so the penetration of antibiotics in the food chain decreases. Because we use our own cell growth media, which is by not killing any animal. So we save one pregnant cow and one calf, because this, in case of chicken muscles, cultivated meat or chicken muscle meat, the chicken is cells are taken from a healthy chicken without killing the animal. We are able to save that chicken. Now we have actually impacted in this process two industries. One is poultry farm industry and another is cow grazing. Now, those two industries collectively, we are able to hit, if it goes to a yearly format, we are able to hit or impact the methane emission of up to 9.2 metric tons per year. And the carbon dioxide emission targeted here is 2.6 million tons, metric tons every year. From a clear meats perspective, what we have been doing in the last 2 years now, we make 1 kg of our meat, we label it as eco meat, through 2.5 liters of Clear X9, our own cell growth media. In this technology, because we have been making small quantities for the last couple of months now, we have been able to save 150 cows till date. We have impacted or we have controlled 60 metric ton of greenhouse emissions till date. And coming back to the procurement time, if you guys remember, I had mentioned clearly that in the initial, the fetal bovine serum for any routine cell culture setup or R&D setup requires at least 6 months of procurement time. But because this is all in-house, and I think every company has to do it ultimately to make sense to the market, the procurement time has decreased from 60 days to 7 days right now. And as I mentioned earlier, also because of the method that we've adopted, we have been able to bring down the price of the industrial POC to somewhere around 2000 INR per kg. So the methods, the before going, so understanding the technology and also understanding the methods that are associated here, it is a longer vision impact that cultivated meat industry envisages to achieve. And there is we have to take a balance between what all ingredients are required and how they are grown and what will be the impact and product impact of it. With this, I will take a pause. I have tried my best to share some glimpse from the lines of sustainability, some glimpse from the lines of the science, a brief science about it, giving you guys a brief understanding of this concept and trying my best to give you a perspective of how lab-based meat is more. I would not. I would, if the right word is more safer and more controlled. It does not use any antibiotics. Does not use any animal killing, and how it is different from plant-based alternatives like soya chunks and likewise, and why there is a demand to it. And this is my LinkedIn QR code. If you want, guys want to connect to me, I am always open to connections on LinkedIn. And with this, I will take a pause, and I'm open to questions from your side.
Um, Thank you, Doc. I mean, before we get into question and answers, and trust we have excellent question and answers, which Samiksha will, speak later on. If you can give the glimpse on how cultured meat is, the science and technology of cultured meat, in the sense that what happens, what comes first, how the stem is, cultivated, and what is the process. So can you give some idea on the process of cultivated meat?
I will again repeat that slide. There was let me share that slide.
And there are some excellent questions so around that slide. So that's the reason why I asked this as a precursor question.
Yeah, I will just quickly go again to the technology slide.
And Samiksha, if you can then take up the
Sure, ma'am. Yes, certainly.
requesting all to keep on.
See, when you look at the process of making cell-based meat or cultivated meat, as I mentioned in one of the slides, it is actually identifying, the whole technology lies in identifying the mass of cell, identifying the cell bank, how that cells will multiply in how much time, what energy source these cells require, and how they are handled in the whole process. Ideally, when you speak of, let me take an example of chicken muscle itself. Now, these chicken muscle are normally multiply. This is known as passage life of these cells, or the multiplication or doubling time of these cells. Normally, chicken muscle cells have a doubling time of somewhere around 28 to 36 hours, depending on the type of cell, the resource, and the nutrition being provided. In that process, once again, yeah, in that process, what happens is, where is that? slide. Yeah. 1 minute. Yeah. So in that process, the cell line identification plays a very vital role in initiating the process, because when you speak of taking a biopsy, there are pool of cells from the upper layer now these are epithelial cells, to how these cells are inside the muscle area, the nerves, the blood cells, and different cell types to it. Now, the method here is to actually identify and separate all these cell types. Let these cell types first stabilize in a controlled cell culture setup in a laminar hood. And after identifying these cells, identify the right nutrition that has to be provided to these cells so that they can multiply in a growth format. When you speak of growing these cells or identifying these cells normally take somewhere around six, for us in our case, it took us somewhere around 6 to 9 months to actually identify specific cell types from the chicken biopsy that we took. We took a muscle chicken biopsy. After identification, these cells were actually grown in a bioreactor format. I used a very basic image just to give you guys the perspective that how things work. Now, this bioreactor, if you look closely, is actually using a pool of cell growth media or nutrition. The cells are continuously rotated. There is a pH is maintained, temperature is maintained. Even you will be surprised. Water plays a very important role on how cells behave and how output is controlled. In our lab, we have to use somewhere around 16 to 18 ohms of double distilled water. So, and there are certain salt concentrations to it. If it is more, the cells will not perform. If it is less, again the pH will not be matched and the cells will not perform. So at ground level, every step is so much controlled to its far best that even a slight mismatch can ultimately lead to uh, decline in the performance of the meat itself, the final product itself. So identifying the cells, the process here is identifying the cells, isolating the specific cell type that we need. In our case, when we did chicken muscle cells, we identified chicken muscle cells specifically, not these stem cells. The image shows stem cells, but there is a reason we do not use stem cells, and that's again technology IP that we have. So identifying specific cells, growing these cells in a controlled environment in a pool of, so it's a mix and match of both cell culture setup and a bioreactor setup, because these bioreactors also have certain limitations, both from an output perspective and also from a cell growth utilization perspective. And then taking out the minced meat out of it. I hope I was able to give you a clarity on how the process works.
Thank you so much. Thank you so much. I think Samiksha, can you take up the question and answers?
Yes, ma'am. Indeed, an very interesting session, sir. Uh, as we go ahead, we have a lot of questions. The first one from Komal is, how does the taste and texture of a lab meat compare to a natural meat?
So it is good you asked this question, and we need to realize that I don't know how much of you guys are non-vegetarians in the population, and I don't want to hurt anyone's sentiment on those lines. As I mentioned in one of these slides, that there are five parameters for any acceptance of food item: taste, texture, smell, nutrition, and price. Now, these five parameters play a very pivotal role in acceptance of cultivated meat or lab-grown meat, the meat that we make in lab. From a taste perspective, uh, there are certain nutritional ingredients that differentiate animal cells from plant cells, and these nutritional ingredients play a very uh, acceptance rate parameter for food items, and this is something which non-vegetarians are actually looking for. So there is a specific taste bud activation that happens when we eat animal-based meat. From a texture perspective, yes, there are certain textures that these cells, because of their adherence to each other, are able to achieve, which plant cells are unable to achieve. So there is a difference between plant cells and animal cells itself. Now, these textures is again something which, as humans, our taste buds are more inclined for acceptance. And this is why when this is this was the one of the main reasons we started with muscles, because obtaining that texture from muscle cells is slightly easier as compared to other cell types. So and I did not mention here, but the meat that we make is actually muscle meat, muscle keema that we make. We are not able to achieve, as of now, there is no technology to actually achieve a full 3D structure out of it, a chicken leg piece. But what we can achieve are minced masses or masses of cells or pools of cell, and that taste and give the right texture as per our expectation. And this has been the feedback we have done. I think more than 20 plus in-house tasting events till date, and the feedback has been phenomenal. Just to give you perspective again, we whenever we showcase our product in any group of audiences, we have to actually stop giving them meat. We normally have an idea of giving one 10 gram, 20 gram meat for people to try it out, but they start eating it non-stop. We have to control that request or demand for that. The second example, recent example that happened to us was we were discussing with some distributors to make meat in lab,
कसं वाटतंय
and the whole concept was it does not
कसं वाटतं तिकीट काढायला
require
आपण
any
बोलतो
animal investigation.
Can
It
I
does not
request
involve any.
participant to be on mute?
It does not require any animal killing. People are ready to accept it. I recently met a vegan individual, and she said to me, "Do you make it's without any animal killing? It is raw meat." I said, "Yes, it is raw meat and without any animal killing." She said, "I'm ready to become your customer." I said, "I never thought vegan people would be our customers." The response that I got was something which again surprised me, because I had never thought on those lines. She gave me a perspective that most of these vegan people have turned vegan because they are more inclined towards guilt-free products and do not have an alternative from non-vegetarian nutrition perspective, and that is why they have turned vegan. And that becomes one more target audience that we fit in. So from a consumer acceptance, a lot has to be done on ground, but from a taste acceptance, from our experiences, as you asked for, there has been a lot of positive feedback, and we are, with every passing day, we are working vigorously to identify how this meat would ultimately look and how it should be presented.
Thank you. Hope this
Thank
addresses. Yeah, go ahead, Samiksha.
Yes, thank you, sir. Another question is, sir, could introducing oncogenic properties to cells intended for cultured meat production elevate the challenges in cell line maintenance? And are there any significant concerns and risks associated with this?
I'm surprised this question popped up, because this normally does not happen. That was one of the reasons I was not going that much technical. I thought I might deep dive into the technical format, and half of my audience will be sleeping. But I'm glad I got this question. Yes, it impacts. We have to realize what food products we are making, what is the final output. If you speak of using methods, the routine cell culture methods, these routine cell culture methods result in genomic instabilities. This is one of the prime reasons we do not use stem cells. Uh, Technologically speaking, stem cells are used in this process of making meat because you can easily alter and play with their cellular profile and impact their output, impact the way more cells can be produced in less time, and providing the right nutrition. This is technologically speaking, and yes, many companies are doing that. But from a food grade perspective, we have to realize what we are making. There's a difference between lab cell culture and lab cells that are being used for human consumption. And there is a thin line of difference. This thin line of difference has to be maintained before it gets accepted for any food item being used for human consumption. Because if I go by the question, you guys might be having a biotech background, and you might understand it perfectly if I use these deep words. Stem cells are induced with SV40 virus so that they can multiply and give the right output, or more controlled output in the longer run, and more cellular viability. But if these SV40 viruses are induced in the food chain, from a research perspective, it makes sense. You can do multiple research parameters. You can understand biological pathways. You can figure out drug discovery modules. But from a human consumption, inducing SV40, a virus which cannot be killed in the human food chain, will impact the food process. Let me give you a first hand experience. In my PhD days, we used to handle SV40 virus. and a small exposure once in every 7 days, only aerial exposure, aerosolar exposure, not even eating it. These small aerosolar exposure once every 7 days for continuous 6 months results in small lumps on the body. Now, these lumps have the potential to become cancerous. So you have to realize that what we do actually in a cellular cell culture setup from a research perspective has to be different from what we do it in a food tech perspective. And this thin line has to be maintained. That is why the technology, yes, it seems simple. It seems a very basic technology when it comes to growing cells in lab, multiplying cells in lab, providing the nutrition. But you have to maintain the genomic stability of these cells. And that is something which we also discuss or share with everyone, that next generation sequencing or genomic profile validation should be made mandatory for such products before they get launched into the market.
Thank you, sir. Going ahead, sir, another question is the digestion time for normal meat and for a processed meat same or similar, or is there any differences in the processing time?
Again, glad you asked this question. When you speak of, again, in my first couple of slides, I clearly gave a hint that when you speak of meat, it is actually mass of cells. The meat that is obtained traditionally is actually a pool of mass of cells of multiple cell types. There is fat, there is neurons, there are muscles, there are epithelial cells. But when you speak of cultivated meat, which is grown in a controlled environment, as of now, the technology limits us to only few cell types in one pool. So the starting material and the end product are same in both the cases, but the methods are different. So digestion time will always remain the same for both the products because the starting material is always cells. For that, in case the traditional meat, or in case of cultivated meat, the starting material is always cells, and the end product is always meat. The only difference is the method how they are multiplied.
Got it, sir. Thank you for answering. Another is how to convert this cell cultured meat into a production line and how is the quality management assured?
So I shared a brief. So my slide, that slide might also be being presentable here. How cells are extracted. So there are from a production line setup. I did not show that here. There are three steps to making meat, and the production line looks as if there are three rooms. Number one room will be responsible for continuous supply or maintenance of cell banks or cell lines. Number two is growing these cells, prepping them or preparing them for mass production. Number three room is taking these cells from these prep area to bioreactors or scaffolds, whatever is the final product that we are looking for, and growing these cells in these bioreactors or scaffolds so that you get the final product. So it looks simplified, but it is more controlled and more, I would say, managed at every level. And when you speak of QC, the second concern, the quality checks, because it gets easily defined into these three rooms. I'm giving only three rooms, but it has multiple fractions to it. So in a simplified version, I'm giving only three rooms. In these three rooms, if you speak of, as they are interconnected, and there are certain SOPs that need to be followed before one step is taken to the next step. So the quality check becomes easy in a cultivated meat setup as compared to traditional meat. Let me give you an example again here. If you look at, if we eat chicken, you, as a consumer, you do not know the origin of this chicken, from which poultry farm it was purchased, who bought it, on which car number or which truck number it was transported, which cage number it was kept, what type of feed it was given. As a consumer, you are not at all aware of these parameters, which play a very vital role in the nutritional value of these cells. You're not even aware if this chicken was actually injected with any hormones to make it more fluffy, to make it more bulky, or it is, this is a natural bulky chicken that you are looking at. The redness in the chicken, is it because of the hormones or antibiotics that have been used, or it is a coloration? So as a consumer, these traceabilities are not possible from the line of these factory farms or poultry farms. But when you look at cultivated meat, because the cells, when they were taken, the chicken was identified. Yes, you can identify the source, or you can maintain a record from which poultry farm that was taken, or it was all in-house reared. The cells are continuously monitored because these cells require continuous validation of their genomic profile, continuous validation of their cellular viability before they pass to next step. The preparation of cells has another SOPs from, again, a cell culture perspective, identifying the performance and the health of these cells, the nutritional value of these cells. The fourth one, the third one, that is how these cells are again form a mass out of it. You have certain parameters which are key parameters to identifying the genomic stability, doing NGS data analysis, doing Western blot analysis, doing real-time PCR analysis, curating the performance of these cells, mass of cells, identifying how many. So in the process of making cells, which type of cell growth media was used, what type of ingredients this cell growth media had, was this food grade or not, was this in a controlled environment or not, was the aerosol controlled or not, because any slight change in the aerosol itself, as I mentioned earlier, the water itself plays a very vital role on how cells behave. I can give you numerous examples from my experience that there was a slight leakage. Whenever there is a rainy season, the chances of contamination in cell culture rise. There is a mycobacterium contamination. So these slight changes actually impact the output of the product. So it is easy to do a QC in a cultivated meat setup rather than in a traditional meat factory. So it becomes more controlled and more hygienic in the longer run.
Thank you for that detailed explanation, sir. Uh, then we come as, can cultivated meat be customized to meet the dietary needs of specific populations, such as geriatric individuals or those with lifestyle diseases?
Yes, and that is something that fits into my personal vision also. The idea of doing cultivated meat, we never thought of it as making final product, and that is one of the visions of Clear Meat, that is feeding the world, feeding the right product to the world. And our vision fits in line of identifying sustainable solutions to the food industry. Uh, the intention here is to make meat. I'm giving you an example, which we are already working parallelly and identifying. So these are all theories that we are working on because it is possible in a cultivated meat setup, because it is controlled from 0 to 100. You can manage and perform the right nutritional expectations of the end product. You can control the output of the product. This is something which is personally close to me. Is for diabetic people, they type 1 diabetic people, they require insulin, regular insulin injections, which is invasive method. Now, in our case, the vision here is that our meat should be able to make natural insulin, so that these individuals, which require insulin from outside their body, need not utilize any non-invasive or invasive methods for insulin injections. They take the meat and then they automatically get the insulin. This is a hypothetical projection, and just to give you an example, it is possible and it is achievable on the lines, and we are already doing something on that format.
Great. Thank you, sir. Next is a question from Rahul. You spoke about how a muscle cell can be used for making a chicken muscle cell line, but the muscle cell line can obviously only make chicken muscle cells. So, in cases of various meats, there are various layers like fat, muscle, how, and they are arranged in a typical certain way to give that particular effect. How can that be achieved in a lab grown meat?
It's a very valid question and something which every company out there is trying to achieve, even for us. So currently, what we or other players are doing is making meat which is of only one cell type. The prime reason to that is the limitations of cell growth media being utilized for one cell type. As you rightly mentioned, there are multiple cells to it, so different cell has different nutritional expectation from the source of cell growth media. Now, because these different cells have different stability or doubling time, different nutritional expectations, they are able to. Currently, it is slightly tedious for us to make a pool of these multi factors and grow them in one structure. And that is one of the prime reasons in one of the slides I mentioned that as of now, people are only able to make minced meat or chicken keema or keema for say, be it chicken or beef. And we are now working, or most of these companies are now working in creating these multi cell line type setups, where you can achieve these 3D structures, where you can achieve these slices which show you the right layer of fat, which show you the right layer of muscles, which show you different cell types. So currently it is not possible, but yes, technology is heading towards making it possible. And to give you a perspective from Clear Meat's lens, uh, when I speak of cell growth media, every cell line, as I mentioned, every cell type has a specific growth nutrition requirement. So that becomes one of the main reason why multiple cells cannot be grown in one dish. But what we at Clearmeat are trying to do is we have created a cell growth media which is uniform to all cell types. So you can grow three different cells in one dish using our cell growth media. So that is achievable, but yes, it's still in an early stage. But you will soon see these products.
So you mentioned about your experience with a vegan individual. A related question here for you is, can you draw a comparison between between the meat, the natural meat, cultured meat, and plant-based analogs based on various properties like cost, texture, taste, etc.
I was thinking you will ask me to put a parity on the basis of its nutritional component, but let me give you a perspective from all the factors that I can think of. From a taste perspective, for a non-vegetarian who is looking for meat, plant-based do not suffice the requirement. They do not activate the taste buds, but meat made through methods that we spoke of is able to match that taste bud activation. So that is one difference. From a price, yes, plant-based meat is slightly cheaper as compared to animal-based, as we speak of right now. But as the demand rises, as there is more development from the product lines size, as we get more consumer feedback, the prices will decline. So that is how a how ingredients are used in a circular format, how continuous the supply happens, and how you are able to take charge. So price, I'm pretty confident in the longer run, it will become much more cheaper as the demand rises. From a nutritional perspective, there are certain limitations. I am not in negativity of complete plant-based industry, but there are certain protein expectations from animal meat, and you have to realize that because of these protein expectations from an animal meat, which plant meat cannot give, that becomes one of the prime reasons or differentiators of animal meat from a plant-based meat. And that is one of the reasons why, as I speak of 80% individuals or humans prefer meat globally, their preference is always meat or a product which has animal meat to it. So taste wise, texture wise, there are products like there is jackfruit that is used to give the texture to plant based alternatives, but there is a sweetness in jackfruit which animal based does not have. That becomes one of the inhibitors for acceptance of these products. So there are differences, there are huge differences between these two categories, but yes, the target market and the expectations are also different. So it depends on the end user how he wants to eat his food. That defines the acceptance of the product.
Thank you, sir. And are there any specific shelf life and storage conditions that need to be achieved for cultivated meat?
Yes. Currently, the methods that have been put in place for industrial POC have minus 20 shelf life, minus 20-degree storage capacity. Uh, because again, it is in a controlled format, you are able to manage the shelf life of the product. Because there are someone you asked me the QC format, the quality checks in the system. Because it is checked at every level, you are able to increase the life of the product. We did an internal analysis and we realized that for any meat, traditional meat that is bought outside in the market, the shelf life is not more than 7 days. But in our case, it can last up to 6 months. The only reason was because in our case, we were able to control the production line. We were able to do quality checks at every level and find out the traceability of ingredients and outputs at every level. That impacted the shelf life. But in that case, in the traditional meat format, it is slightly tedious. That is why it has a lower shelf life.
Another question seeks your opinion, sir. Is use of 3D bioprinters or additive manufacturing, like addition of suitable additives, would that make these products more acceptable?
Uh, I would not deny that, but I would at the same time not agree to that, because it is too early to comment on those lines. Because yes, we need 3D structures, we need additives. There are companies which are actually using plant-based fat to provide the taste expectation to cultured meat. So these add-ons are required in the process of making meat, but this is not an immediate demand, I would say. This is again from my own personal experience. I've realized that uh, obtaining a foolproof food product cannot be done overnight. It requires generations of research and feedback, consumer feedback, before the final product is achieved as per the consumer's acceptance. Currently, the technology that is available out there, it is slightly difficult to obtain these 3D structures or implement these 3D technology in a cell culture setup. The core reason to that is, I did not dive into it, but I think I should give you a brief. Uh, from a technology perspective, bioreactors are utilized in any animal cell culture for mass production. These bioreactors, when you speak of animal bioreactors, as on date, more than 10 liters of capacity of bioreactors are not manufactured, because of the reason that cells, when there is increase in the capacity of these bioreactors, the performance of cells decreases. And cells are like organisms, they tend to identify the change in their environment. So you have to be very polite in how you handle them. So there is a technological limitation of these bioreactors itself that impacts the output. So yes, there are certain upgradations required, but there are certain technological limitations also. I think that will be achieved as the demand rises and as more brains are put into it.
Great. Thank you so much. Another is from a health concern point of view. Are there any long-term health effects of consuming cultivated meat, particularly with regards to potential allergenicity or other safety issues? And how do you plan on addressing them?
See, that's the beauty of cultivated meat, because you are able to take charge of all the input ingredients. You can manage the health impact. You can quantify the health impact and make products which are more consumer friendly. I gave an example of zoonotic diseases. Because these cells need continuous monitoring from various ground parameters like cellular viability, genomic stability, housekeeping genes performance, the status of proteins, carbohydrates, and likewise the vitamins. Because there is a traceability at every step. We at the cultured meat in the cultured meat industry, you are able to take charge of what will be the final product, how nutritious it will be, how less allergic it will be, or what impact it can do, and how you can control that, what factors you can remove from this meat so that it becomes more acceptable to the consumer. So from a cultivated meat setup, it becomes easy to address and answer these concerns as the demand increases. But from a traditional meat, as I will again say, it is pretty difficult because there is no outlined QC. It is not possible. I'm not blaming it, but it is not possible to manage those QCs in a traditional meat, but it is possible to do it in a cultivated meat setup.
Got it, sir. Uh, can this meat be certified halal or kosher certified?
Yes, it has already been certified. Israel, there are bodies in Israel and Emiratis who have certified it. The reason they have been able to do it, so it's good. Discussions are happening on those lines, and I'm getting more to experience from you guys, the perspective of the consumer, because we hardly get these exposures in our journey. From a halal and kosher perspective, because as I mentioned about the four pillars, the cell lines, the methods, the cell growth media, and the licensing part, the origin of cell lines identifies the final product's profile. So it has been labeled, it has been given certification by Israeli government and the Emiratis also, that it fits into the halal category because the source of cells is from a halal category. Now it becomes easy, again, same line. It becomes easy to control the output, to keep a quality check on the performance of the output, making it more consumer friendly.
Got it, sir. Uh, then some from a cost perspective, is there a cost difference between natural, I mean, what is the cost difference between natural meat and cultivated meat and fat?
As on date, there is a huge difference between traditional meat and lab grown meat. I'm taking an example of chicken itself because the audience, I feel the audience might be able to better understand. If you go out and buy a chicken, raw chicken, it costs somewhere around 200, 150 to 250 INR per kilogram. And if you go out and, in case if we start selling it in the market, the chicken that we will sell at somewhere around 2000 INR per kilogram. There is a price difference of more than 100%. And this is because of the technological or output challenges that are associated with the demand supply. But when you speak of the processed meat industries, when I speak of processed meat industries, these are chicken sausages that you buy, predefined packaged food. Because packaged food is already, as Simran ma'am also mentions in many seminars, ultra processed and processed meat. This packaged food utilizes so much technology to it. The price point increases on those lines, and we are able to achieve the price parity when it comes to the processed food industry. And that is our immediate target setup to fit in at least one of the criteria of consumer acceptance, that is processed food industry. Once it has an acceptance from the consumer, from the processed industry setup, we will slightly, because of the demand supply again, and because of technological advances, we will be able to bring down the price as the demand increases.
Thank you, sir. As we come to the conclusion of this session, we have a futuristic question here. Do you see cultured meat being ready or being used in space travel in the future?
There are already demands for it. Let me put it this way: space travel is one of the reasons that many companies in US have established to cater to the demand. NASA. From an Indian perspective, I don't know if this is the right channel to discuss it, but the bodies in India are already in touch with us to make such products available for both the army as well as space. So you guys are thinking, but individuals are already way ahead on the lines and trying to make it possible in the coming years.
Great, sir. Thank you so much for those insights. One last question that popped up. Uh, what is the timeline for the production of a cultivated meat, right from the biopsy process to getting the final product?
So in our case, as I did not give a brief about it, a specific reason. I thought the audience might sleep in my discussion, but I'm glad it followed in the QA and makes sense to the whole thought process and the vision. The process of identifying cell lines from a biopsy is somewhere minimum 6 to 9 months to maximum 2 years from a perspective of technology and industrial scale up. Once these cell banks are created, it takes another 30 days to prep these cells. So before they go into high throughput manufacturing, and another 15 to 20 days to get an output out of it. From a numbers perspective, for us, I will give you an example. In our case, it takes us 45 days after creating the cell line or cell bank. It takes us 45 days to manufacture 1 kilogram of meat. So this is a small proof of concept that we do. And yes, again, as there is an infrastructural upgrade, the production line will increase.
Great. Thank you so much, sir. Indeed, an insight-packed Q&A session that we had. The knowledge that was shared by you will definitely help many of the budding entrepreneurs in our audience to move towards this line. Thank you, sir. And as we move towards the conclusion, I would like to request Mr. Ritesh Mathur, Vice President of AFSCI Mumbai chapter, to please uh, give the concluding address for our first inaugural session of the Edu series.
Hey, thank you, Samiksha. Thank you, Dr. Siddharth, sir. Wonderful section. I just loved it. I just heard the word cultivate meat and before this. The way you explained it, the depth and horizontal way was really amazing. I love to see that more than 100 participants, almost like 95, 96 it touched it. Was amazing. And usually like in our previous experience, we usually had like the session go for like an hour and the question answer go for like 2, 3 minutes. This time was the other way around. So I could see a lot of interest among the student population or people who are attending this session. I could see a lot of people are asking these questions. A lot of buzzwords, a lot of things people got interested. Uh, Amazing. Like I didn't expect that, very honestly didn't expect there's too much questions to be coming in, but the way people showed interest was amazing. This really indicate this is a very hot topic and more people will be bothering you on this. So wonderful session. Just loved it. The way the depth you had on the session, they we enlightened the entire session on the culture meet was amazing. I also want to thank you all the students who or all the participants who took a time on their Saturday and come for this session. Attending this is superb. This really showed the seriousness, the gravity you had for the subjects. Amazing. Thank you so much. And we keep having all this. We having sessions at every second and fourth Saturday. So please do join us for this session. The next session will be on the world technology requirement for the food emerging preparedness by Dr. Vikas. Yeah, there's the next session. It's gonna be happening. I also want to thank all my colleagues from FST, right from our president Chenmai ma'am to Jyoti, our vice secretary, to everybody in the entire FST, and of course the CSMB, right from Nilesh Lede to Umesh to Prabhu sir, everyone in the team. I want to thank you for this wonderful session. For this, I also want to take a couple of minutes to talk about the FST things. Samisha, can you go on FST slides if you have prepared those? And I would request everybody who is here. Chinma did touch base with the in the morning session, like in the starting session. She just touch based on FST and requesting everyone to be a part of the FST. And those who are not the member, please become member. And especially the students who are very keen for such sessions, I will request them to be a part of this FST. So we do a lot of such events in FST, like and we always think about कि okay, मुझे क्या मिलने वाला है FST के अंदर. So here are some example. Like of course you can get a networking, you can get a chance to present a paper, you can be a part of this such events, you can attend these sessions. There's a lot of award program we do it, a lot of academic things can happen, a lot of mentorship which senior people from FST can mentor you as a student to them. We can support in all the MSME events which Government of India propose or FSI shares. We can share with you. You can be a part of the entire event or all entire value chain. You can be part of that. Right. So a lot of events, a lot of such things. You be part of this to student directly. Otherwise, you may miss it if you don't become a member of FST. Right. So
thanks, Ritesh.
To be a
and
member
thanks,
of this.
Chinmay. Thanks, Samita. It was a wonderful session. Thank you very much.
Thank you, Konika sir. We have more slides to present, Samiksha. Yeah, these are some initiatives of the FST which we want to share very quick base. So we are always a partner with the event like IFCON and IFCOS, which are done by FST home event. We always being a part with the or regular summits. We always being a part of the event like a FI or Annapurna or a USP, US Chickpea Council, USP Soybean Associations, other dairy events which are done by either Colimesi or Inform Markets, many other companies who does that. And we do a lot of events. Few of them are flagship event could be like Nutrition Week, World Food Day, World Food Safety Day, right? Or Analysis Day. So we keep on doing such sessions. Then this is Food Tech Sherpa. It's the new initiative of the current year. And we are doing a lot of such interesting sessions. So I would request everybody from all student community to come for this session and attend these sessions. And let me know knowledge sharing works. Yeah, so this is the next session. So we'll see you on second Saturday of the May, uh, on very interesting topic on world technology requirement for the food emerging preparedness. Very interesting topic. This is what we're talking about in today's world. So yeah, looking forward for this session. Over to Samiksha for you for the concluding anything you want to say over and about this.
I thank you, Ritesh, for this wonderful closing remark. I just want to tell all the students that apart from organizing technical sessions, we also support in terms of helping you for preparedness for developing soft skill, preparing for interviews, connecting you with the industry visits and number of things. So be part of the association wherein you get connected with 8,000 plus food professionals across India, and and be part of the journey. I guess, I mean, we are ready to pass on the baton to you when it comes to, taking the association further. So thank you so much, and over to you, Samiksha.
Indeed, thanks to all the office bearers and the LEC for uh, coming up with the concept of this wonderful session. Thank you to our speaker also for sharing all the insights and knowledge that he shared during the session. We look forward to seeing you in a future session. Before we go, I request all the participants to please pitch in their feedback through the link that is provided in the chat box. We have a link that is shared here by Nidhi in the chat box. Please go ahead and give us your feedback on what more that you would like to learn about in our upcoming sessions.
And
With that,
yeah, I think with that, with a good round of applause for Dr. Siddharth, we conclude the session we conclude this first series. Thank you so much, Dr. Siddharth, for the fantastic inaugural of this series.
Thank you, ma'am. My pleasure.
Yeah. Take care. Bye. Have a wonderful weekend.
thank you all. Conclude the session.